and today I'm going to share uh, my last project that happens in my PhD, and we will, to make sense, preprint in the coming months. And yeah, so in short, the effort in my PhD, also my maybe 3-4 year postdoc, will be focused on to building AI and machine learning methods that can advance sciences, natural sciences. And nowadays, we know foundation models is a very hot topic. So we're thinking of, can we build a foundation model? Can we build an AI scientist? And on this kind of directions. And so this paradigm is borrowed from the famous paper called Segment-Anderson. And they kind of to divide the development of a foundation model into 3 parts. So there's model development, there's a data curation, engineering, or whatever, and also a carefully designed, maybe portable pre-training task to train the models. Okay, and I spent some time on all these 3 parts in my PhD. So today we're focused on the data part, because we know nowadays the people are kind of focusing on, kind of realize that the quality of your pre-training data really matters. Like people are using like only textbooks and train the model, and the model can perform very well in solving programming tasks. So today we're going to focus on, can we use the modern AI and ML methods that can help us to create a better science data that can use potentially for training a very generalizable AI scientist. Okay, so this project is about the domain of single cell biology. So we're going to talk about what is single cell biology and what is the data looks like, and what is the interesting tasks that we can play with in this area. So when we talk about single cell biology, maybe 80 or 90% of the time we're talking about the single cell RNA sequence. It's like when we talk about language models, maybe 80% we're referring to transformers. Okay, so what is single cell RNA-seq? So we take human as an example. Suppose like there's a patient, they're in the hospital, and we want to know what certain changes in a specific tissue. Maybe it's brain, maybe it's lung or whatever. And you can take a part of it and you do the sequencing. And so through all this kind of wet lab equipment and procedures, the final data that we have in our hand is essentially a tablet data. So it's a high dimensional, tens of thousands dimensions, and also a highly sparse data. So the data looks like as the second rows in this figure. So it's a cell by gene count matrices. And so each element, each entry in this matrix represent how many of that gene are expressed in the cell or are measured in the cell. So essentially you will have like 20K different types of genes. And these genes some of them we're already very familiar with, known for certain biology functions. Some of them we are still trying to understand them. And you get cells. So you can have like, so nowadays people are talking about millions of cells. So you can say it's a very large matrix, and there's a lot of zeros in there. So it's a very sparse matrices. So this is the major ingredient for a single cell sequencing data. And we can use this kind of data to do a lot of things. Yes, so for instance, you can use this kind of data to develop new therapeutics, new therapy. So a major idea is like you have a cell. Well, it's not necessarily need to be an animal or so on. You can in the lab you can have a device that can, grow cells and do some experiments on it. And you can give it a different molecules. You can give it different drugs. You can give it a different chemical environments. We call them perturbations and conversions. And you can then observe that how this cell will change. Like maybe some of them are malfunctioning, and you're adding certain molecules into this environment, and the cell will return functional and so on. So this paper, which is originally submitted in Science, but it got rejected, so they finally end up in the molecular system biology this year. And so essentially the idea is that you use autoencoder on that tablet data, on that large sparse matrix, and they try to learn disentangled representations. So part of them are, represent what kind of modifications, what kind of perturbations you've added to the environment, to the cells, and parts of them are just some random learnable events. And this paper is basically to to make the latent space composable, and so it can be interpretable, and therefore you can learn what kind of combinations, what kind of the drug combinations will cause what kind of effect. It's essentially this what this paper is working on. And you can navigate in the latent space. You can try to find a better solutions, a better treatment, and also you can do some predictive tasks, such as to predict the outcomes of adding certain drugs to the environment. And another very interesting application in single cell biology nowadays is like we can use this kind of data to understand certain therapy. For instance, we know like mRNA vaccines is definitely a very promising menu for the future treatment for diseases. Like our mRNA therapeutics is for sure a very good promising candidate. So this kind of things is all belong to what we call gene therapy or cell therapy. So essentially, it's like you can take parts of your cells, like T cells. T cells is the type of cell that can kill tumor cells. So you can take your T cells out of your body and replicate it maybe 1 million times, and then put these T cells back, and then these T cells will, kill the tumor cells for the patient. And this is a very rough description of the cell therapy. So what AI and ML can do is in it. So at each time step of the treatment, you can take the sample from the patient. You can sequence thousands of cells. You can get a very sparse matrix for represent each time step, along with the conditions, the treatments, also the patient's healthy status, and so on. And you can use all this kind of information, try to understand, and also help you to refine the design of your experiments. So this is also a very important application field that AI can help with single cell RNA sequencing data. And if you're maybe interested in this kind of topics in the future, there's a well prepared paper list on the GitHub to give you a collection of the recent noticeable papers, like the data collections we call atlas, like how to visualize them, and how to design, maybe how to design a metric learning to learn like the distance between different cells, and how to do a dimension reduction, how to do a better clustering, how to do the representation learning. So essentially that GitHub have collections of this kind of topics. And there's also how to say, a course that I highly recommend if you're interested in this kind of if you're interested in general machine learning for computation biology, you can watch Manoli's lessons. And so he's a professor in MIT and also World Institute, and he teach this classes every year. And there's also some very famous traders. You can take a look at their traders. So I will share the slides to the mailing list after this talk. And so besides this interesting promising applications problems, essentially data quality is kind of in general, data quality is very important in particular in healthcare, in biology for sure. And so one of the famous issue with single cell biology, with single cell RNA sequencing data, is called batch effect, or a more familiar term would be heterogeneity in your data. So, so for instance, you get a lot of matrices. So each matrices maybe you collect from one sample, and you want to make use of all these kind of matrices to do an integrative analysis, to do the integrative benchmarking also. So first you need to do a general pipeline is to do a projection at first. So as shown in the middle of the graph. And what we usually observe is that cells from different batches, from different samples, they usually tend to be clustered together. So essentially, all the cells from the same sample look similar, instead of all the cells have same functions, all the cells have same types of same biological characteristics looks similar. So what we want to do is we want to remove this kind of batch effect. We want cells that have same functions to be close, to be together in the embedding space, instead of cells from the same person, from the same donors to be the close to each other. Okay, so there's a lot of opening questions, unresolved issues in this direction. So what we want to solve in this project is two parts. So first, we want to understand insights. So we want to understand what kind of the results, what kind of the factors is the cause to make such things happen, to cause this kind of batch effect. And we want to learn from the data, and also we want to learn this kind of knowledge should be transferable, to be transferred to other different data, different types of organs, different types of experiment conditions. And so based on the good understanding of this, we want to design a data engine that can, systematically remove the technical factor, the unwanted technical variance from your data, and we only keep the useful variance in your data. And this is one part. And also, if you can know how this technical variance cause the data to change in what way, you can generate data with novel combinations of your experimental conditions. So this is like two slightly different subdirections. Okay, so there's a lot of existing efforts in this direction. So there's some foundation models that published last month. So there's also a few papers that are benchmarking in this single cell genomics as well. And also there's some papers that are trying to learn a universal cell embeddings, which is like they try to implicitly seize or reduce all this kind of variance on unwanted variance and try to learn a universal cell embeddings across different species. And yeah, so many of them are from Yuri's group, which is one of my postdocs advisors. And for sure, there's a lot of a few works to do. How can we generate synthetic data? Can we generate different kinds of single cell RNA-seq to facilitate some applications, some methodology development? Okay, so in this project we call GCBM, so it stands for concept bottleneck models for a single cell RNA-seq data. Concept bottleneck model is a paper that from Personnel's group. So essentially in that paper, they kind of design interpreter model for images, but we kind of use this idea in single cell biology stuff. And so retrace the framework that is proposed by the segment Anderson. There's 3 modules to build foundation models in science. And so we start by looking at the data. So recall our motivations. We want to have a data that can record different experiment conditions. So essentially we chose this one. So it's focused on the human lung, and it has like over 100 people. And it has very diverse protocols. And also the people are very different. Like there's smokers, there's non-smokers, healthy people, people have lung cancers. There's also different races, different genders, and also different body weight, how to say, as well. And yeah, so this is kind of the atlas where we're used to train on. And there's a lot of, also this atlas, they have a lot of technical and demographic variance, which we identify as the variance that we don't want, like smokings, like this health status, like what kind of software and hardware you're using to do genome sequencing, and the demographic, like the personal information for the donors. We have yeah. So if we do some clustering on this kind of data set, we will notice that so each point in this embedding space is a cell, and we will notice, and it's colored by the different experiment conditions, and you will notice that cells that are from the same experiment conditions are kind of the same to be close to each other. This applies to nearly all the experiment conditions. Instead of their types, so we use cell types to represent a cell's functions. So if we use an example of the images, it's like you have like let's say suppose you have 2 phones. One of it is iPhone, another is maybe from Sony. I don't know what Sony's phone is called, but yeah, the Sony phone. And you take a lot of pictures. Use either of the use both of the phones, and now you do the embeddings of all the pictures you're taking, and you want to embedding space to have the embeddings of the dogs, no matter which phone you're using. You want all the dog pictures to be to be close to each other, but you want you don't want to tell which phone the photo is taken from based on its embeddings. So what is batch effect in the single cell genomics is like we want cells that have same functions to be close to each other in the embedding space, but we don't want to tell where the cell is come from. Does the cell is come from a male donor or female donor? In the downstream analysis, that information is not usually helpful So we want to remove such variance, but only to keep the fact that its functions, the fact of its functions. In this way, we mean cell types. Okay, so the above figure, the cluster represents what if we do with the raw data, and after batch effect removal, we kind of to have a very good clusters, which is similar cells. They are embedded close to each other. Okay, so this is what the data we have in the gene CBM project. So now we're going to talk about the model and the task. Okay, so the model is very simple. The model is just we follow the concept bottleneck.
Can you hear us all right? Michael's just got a yeah. So what's the reason? What's the reason for the existence of batch effect? Is that I don't know, this sequencing machine may have a certain bias or something?
You're right. So the observation for the batch effect is like the expression level of certain genes will change under different conditions. So for instance, maybe under some measurements, under one is a specific measurement protocol, maybe certain genes, let's say gene 1, they will have 0 observations across all the cells, but that's not the true. That's not the fact. So different experiment protocols, different like say different instruments, they will cause the observation will become slightly different from each other. So this is the cause of batch effect. This is the outcomes of batch effect.
Yeah. Okay. So my understanding is it's almost like a causal inverse kind of scenario. You have two combining factors. One is this batch effect, and the other one is the true underlying whatever data generating source you have. So you're trying to say, can we isolate the batch effect and only try to model the true data generating process? Is that correct?
Yes. Yeah. But a slightly difference is that we don't use a causal model. We use a data-driven method called concept bottlenecks. So essentially, we want to remove this variance that is irrelevant with downstream analysis, and we want to keep the useful variance for the data.
Another question: how do you know you can remove this batch effect? Essentially,
so in single cells, usually people do clusterings. So you let's say you remove batch effect no matter on the data domain or on the embedding domain, you do embeddings on your you do clustering on your embeddings, and then you have how to say, you have data cell types for each cell. So essentially, the cell type represents their functions. So you take a look at the cluster, and what we're expecting is we want cells that have similar functions that are be clustered together. So there's a lot of measures on the clustering, like there's a lot of measure on the clustering, like maybe something on the nearest graphs, the consistency, yeah.
Right. So you're saying your data live on a very high dimensional space, and if you find a way to reduce the dimensionality smartly, then you can get rid of the batch effect. Is that right?
I'm not sure if I understand you correctly, but the general protocol is like we have this high dimension data, like 20K dimensions, and we usually use like PCA or we use MLP. We project them into like 50 dimensions, and you can use some classical methods like canonical component analysis that can remove this kind of variance or to reduce this kind of variance from this embeddings. And then you measure how well your method works.
Okay, thanks.
Okay. Okay, so next we're thank you for questions. So next we're talking about model and task. The model is very simple. We follow Percy's paper, concept bottleneck models. Just use autoencoders. Oh no, well, I think in that paper they use convolutional neural networks because it works on the images. But yeah, we follow previous literature on single cell biology. And so a lot of papers, for sure, nowadays people are using transformers. And yeah, and it's still in debating which architecture works better in single cell biology. But yeah, we use autoencoder because there's a lot of already existing works, so we're quite familiar with these model architectures. And as the same as the concept of bottleneck models, we have two parts in our embeddings: the bottleneck parts and also leakage parts. So instead of the discussion in the leakage in the concept bottlenecks, what we want is like we want the leakage to keep the useful variance, and while the bottlenecks is kind of to take care of the unwanted variance. And the precision task is also very straightforward. We have like three parts of different objectives. The first is like a reconstruction. We want to train the autoencoder, so therefore there's a reconstruction. And what we do, what we can do with this reconstruction objective is like, first we can learn a low dimensional representations, which is the encoder's job, and then we can generate a new or original data that is the decoder's job. And if you are familiar with the concept bottleneck models, you can do some perturbations or manipulations on your embedding space. So in that way, you can generate new data with known concepts. And there's the first objective. And second is to do metadata predictions. So you kind of train the encoder, try it to predict the experimental conditions of each data, of each samples. And yeah, so it can allow us to know the experimental protocols for unseen data, which means the knowledge is transferable. And it can also enable us to do some explicit interventions on the metadata on the logics. And the last part of the objective is to make the embeddings, two parts of the embeddings, to have less dependence from each other. So we use, for instance, you can minimize the mutual information, minimize the info entry loss, and also you can minimize the minimize the dependence use like some measures like we called Hilbert-Schmidt's independence criteria, which is also another loss, differentiable proxy. You can to decorrelate the two random vectors. Okay, and okay, so yeah, so we will save the math part, and we'll just focus on the main idea. And so by such design, the gene CBM can do three different things. The first thing is like you get a new data point, new like large sparse matrices. You can predict what kind of the conditions this sample that's collected from, like in the male, female, smoker, non-smokers, what kind of devices you're using, what kind of which parts you're collecting from. And also, you can use a technique like GradCam to interpret what kind of to assign the importance scores for each of the data, each of your input dimensions, and you can interpret which gene have the most contributions when you make such predictions. And this is the first job that GCVM can do. And the second job it can do is like the same as concept bottleneck models. You can manipulate and you can intervene your latent space in an interpretable way. For instance, you can change the devices, the sequencers, and if you you also change different rates, different conditions, like the sample can be fresh or frozen. It can be male or female. You can change, the bottleneck parts of your embeddings, and you can generate new data. And the third thing is like you can use it as a data engine to integrate a lot of samples together. As we said, we use infoacy, we minimize infoacy to minimize to minimize the visual informations, and so we will use the leakage parts of the embeddings, which we know is kind of very irrelevant with your experiment conditions, and we use that to represent the cells to do the to use it as the representations of the data for downstream analysis. And for sure, we can also to project all the data in the same experiment conditions. This is another way that we haven't tried, but we think it would be very interesting to try. Like we can, instead of to remove the information of the experiments, of your experimental conditions, you can project, you can map all the data into one specific experiment conditions. So this is kind of, you do integration on the data space instead of the embedding space. Okay, so there's some results. The first is like the test accuracy. As we said, the first task is like you give me a sample and I can predict what kind of the metadata, what kind of the experimental conditions the sample is collected from. And our model has two parts, the bottleneck parts, which is denoted as gene CBM-B. So its job is to, to capture all this kind of variance, technical variance. You can see the test accuracy is very high. It's 99.9% something. And the leakage of the gene CBM, the leakage part of the embeddings is try to learn the embeddings that are not relevant with this technical variance. So you can see its test accuracy is the lowest in comparison with all the previous method. So all the preliminary method like UMAP, TSNE, PCA, they're classical dimension reduction method, and we train a linear model on that to do such predictions. And SCVI stands for single cell variational inference, and SCAVI, Scanormer, Harmony, they're previous project that are designed to integrate the single cell data. So you can see our so by explicitly add this to your objective, we show that it can the two parts of embeddings can do their jobs. And you can also apply some GradCam to to identify the genes that are important when you're predicting a certain experiment conditions. And so in this bar plot, we show that when you're predicting a sample is from a male or female donor, essentially it's the genes that are located in your X or Y chromosome. They are the main contributors. Their expressions are the main contributors for the gene sequence model to make such decisions, which are well, in align with what we know. And well, actually, GridCam has also have some issues in the single cell data. It's like one of the issues is instability, so or randomness. So if you run it across different model architectures, you will get some slightly different results. I mean, slightly different importance scores for different types of genes. And so this is one example. And in the manuscript, we provide some more biology interpretations to do that. Some of them are, I would say, either it's false negatives or some new, maybe unknown scientific knowledge. And yeah, so essentially you will see all these genes like exist, PS4Y1 and DDX3Y. They are all the genes that are located on your X or Y chromosome. And so these are known as the main factors when you're predicting the gender. And when you're predicting the smoking status, you will notice that the genes that are responsible for recovery, they're kind of the main factors that will have large influence when you make predictions on the smoking status, because we know that smokers, when you smoke a cigarette, it will cause damage to your lung. So therefore your cells will, form a recovery mechanism, recovery process. So it will have more this kind of genes to be expressed. Yeah. And second,
I'll go back to the architecture page. The neural network architecture. Okay, from way back there. Yeah, this page. So I have several questions. So what is technical meta?
Technical meta is like metadata for the data, like the parameters in this table. So we call them metadata or technical meta. So it's essentially the metadata that records the technical conditions of your sample.
Okay. What's the format of this data? Is just a tabular or what?
Sorry, what's your question?
So I assume every sample have these have one set of data associated with it. Is that correct? For every sample.
We have like in this we have 166 samples. 166 samples collected from 107 people, and there's like 500k cells. So essentially we have 500k data points, and we use 70% for training, 30% for testing. Yeah. So we're predicting the metadata of each cell instead of we're predicting the metadata of each sample.
So back to the architecture page. Well, what is leakage?
Oh, it's a term that is used in concept of bottlenecks. So the leak, probably we need to change the name. But so essentially, there's two parts of the embeddings in our design. The first part is to take care of the metadata prediction. For instance, like there's like 2 dimensions that can represent gender. And the 2 dimensions as a summation of 1, and both of them are. So essentially, they represent the probability of your metadata, and like 0.3 for male, the dimensions, 0.7 for female as that dimension, that's different dimension. And the leakage is the embedding that is learned from the data. And we want the leakage parts to learn the useful variance. We don't, and that is apart from the concept part, the technical meta parts, the concept parts,
okay.
The bottleneck part.
And you design the bottleneck, the dimension of the bottlenecks, right?
The dimension bottlenecks is based on your data. So it's essentially the summation of all the number of categories from all the metadata. So there's maybe 45 or 43 something. And we design the dimensions of the leakage. And in the study, we use 60, and also we try 16. Also, we try 64, which also try 2. We also try 0, but lastly, we stay with 16.
And you mentioned you're doing some extra experiment to help with the decorrelation. If I understand correctly, is that I don't know, is that very expensive in real life?
So actually, go ahead.
Continue.
So we just add another law of terms when you train the data from the beginning. It's like you want to, so bottleneck part and leakage part, you treat them by 2 random vectors. So you want these 2 random vectors to be irrelevant. So we use influence E, and also we try Hilbert-Schmidt independence criteria. These are differentiable laws that can use as proxy to reduce the dependence from between these 2 random variables.
That makes sense. Thanks.
And okay, and we can also So back to the second applications of the model of this framework. You can manipulate the metadata. For instance, you can change the gender of your sample. So for this sample, as shown on the table on the left, it's a male, and it's collected on the 3' sequencing, and we can manipulate, we can intervene it into a female. So okay, let's say we now have a data that is excited for the male. What does its female counterpart looks like? We can have our model enable such modifications. And this kind of things has very good alignment with known biology knowledge. And and last part is we can use it as an integration. You can, they said we can use the leakage parts as the data we use for downstream analysis. So we use the metric that is proposed by the first reference in the bottom. They propose basically two types of metrics. So first is we call the biology conservation. It's like how well your clusters can conserve biology knowledge, like each cell. Cells from same cell types, from the same types, with similar function, they should be clustered together. And batch correction is like how well or how well your clusters, how worse your clusters can reflect the sample ID like which sample it's collected from. So the more mixing your cluster is, the better you correct the batches. So essentially, these are kind of scores, and all the scores are between 0 and 1. The higher is better. So as you can see, our model has the best scores, and as well as visualization as well. Yeah, and right. So essentially, we plot the visualizations of different methods, and you can see our model can have better integrations. And if you dive deeper into specific cell types, for instance, the challenging cell types, you can see our model perform well as well, which means it can separate this very tiny clusters, and well, the original is failed to do so. And also we show that such trained or pre-trained model, it can be transferable to other data collections. Yeah, so we can, if let's say you train on a, it can help you to integrate data on heart, another organ, or on your arm or so on. And we show that first, we show it can predict the experiment conditions of the other without any additional training. It can already use as a plugin predictor to predict the conditions. The only reason we have only like 2 or 3 metadata here because those data sets, they don't have very good how to say, records to record all the experimental data. So the only information they have is like the gender or what kind of kids they're using. So we just test the scores. And also we show that with fine tuning, we can have better we can have better integrator on different data sets as well. It's very, yeah, it's well, when our collaborator first saw this, they just said it's too good to be true. So I think one of the major idea to explain such things is like we explicitly remove the variance, the technical variance, the unwanted variance from this embedded. So this embeddings will have this higher scores or visualization as well. Yeah, it has this very good visualizations. As shown in this. So in this case, it has like over 100 cell types across 100K cells. It's not easy to go to separate cells like this. And there's like a future directions for sure. You can try transformer, larger data, also try multimodality as well. And yeah, so in short, we propose this work. It can do 3 things. So it can predict the meta, it can predict the experimental conditions of your sample, and it can generate new data. It can generate with some unseen combinations of your experimental conditions. And also it's a better integrator. It's a better glue to combine all the data. Yeah, so this is my lead project, and also we have collaborators from other institutions. Okay, thanks for time.